Today we are going to discuss anatomy of storage roots. We have already completed the morphological part as far as taproot and adventitious roots are concerned. So now we will be having the internal structure of few storage roots, amongst which the first one we are going to do today is beetroot. Okay. So the fleshy storage roots become thickened to store food. For example, sweet potato, beetroot, radish, turnip, etc. So where does this food get stored? It is always stored in parenchymatous cells. It means all fleshy roots will be having abundant parenchyma, right? So here food is stored largely in the xylem parenchyma or phloem parenchyma or both. Okay, means during the secondary growth, when secondary xylem and secondary phloem elements are cut, along with that the parenchyma also develops, and if it is a storage root, abundant parenchyma will develop. The food can get stored in the xylem parenchyma only, phloem parenchyma only, or both. So now we are going to discuss the first example as far as the internal structure is concerned. So internal structure of beetroot. Beetroot is known as Beta vulgaris. It's from family Chenopodiaceae. So initially, if we observe the primary structure of this plant, it has two vascular bundles. So it has a diarch condition. It means two xylem patches alternating with two phloem patches. Okay. Then initially the vascular cambium is absent just like other roots, but it gets developed as we have studied in a dicot root, that is there is formation of vascular cambium from conjunctive parenchyma and few cells of pericycle. So that much growth will remain same, but afterwards the activity of cambium is different. Okay, the place of development changes. So it's a kind of anomalous secondary growth which continues after the very first step, that is the formation of vascular cambium. So initially if we see the primary structure, so in primary root, two vascular bundles are present in beetroot. Thus, it is diarch. The secondary vascular elements develop through the activity of vascular cambium, which is anomalous. Okay, only the first ring is normal, Later on, number of rings are added for secondary growth and they are all anomalous. Anomalous means abnormal. So first we are going to define the primary structure. So here the primary structure is typical dicot root type having epidermis, with unicellular hair, root hairs, parenchymatous cortex, endodermis, pericycle and radial vascular bundles. Xylem is diarch and exarch. Exarch means protoxylem is present towards outer side and metaxylem towards center. Normally in dicot roots pith is absent, but here a small pith is present. So pith is small and parenchymatous. Okay, that was about the primary structure. So in detail we are going to discuss secondary structure. Secondary growth in beetroot is anomalous. I told you the formation of the cambia is abnormal in this case. A normal vascular cambium is formed from conjunctive parenchyma and cells of pericycle present opposite to protoxylem. The cambial strips join to form vascular cambium. Further, its activity is abnormal. Okay. So the first step will remain same as we discussed in a dicot root, that there is formation of vascular cambium, because initially in the primary structure cambium is not present. So during the secondary structure, secondary growth, firstly the vascular cambium will develop, and that is in a usual way, that is the cells of conjunctive parenchyma lying in between primary xylem and primary phloem, they become meristematic and they join with the few cells of pericycle present opposite to protoxylem, which also becomes meristematic. So that all the cambial strips, they join to form a functional vascular cambium. Now this vascular cambium should divide to form secondary xylem towards inner side and secondary phloem towards outer side. That is a normal activity of vascular cambium, but it is not so in this case. Here the activity of vascular cambium is abnormal. Okay, so the second step where we discuss the activity of the cambium, that will be a different one in this case. So that we will discuss in the next lecture.